Welcome to the mitosis and meiosis simulation. Today we will compare two different ways for cells to divide, meiosis and mitosis. Cell division is essential to build and maintain living beings, including humans like you. As your AI assistant, I don't have any cells, but I still love to study them. First, click on a lab coat to put it on before starting to work in the lab. The second step is always putting on gloves. When you are ready, click on the glove box to put gloves on. The researchers in this lab have extracted an interesting compound from a yew tree. Your mission is to find out what effect the compound has on dividing cells. Let's get ready to work in the lab. This simulation uses voiceovers. If you don't have audio, click the speaker symbol on the top right of your lab pad to switch the play mode. If you disable the sound effects, the lab pad will slide up whenever there are instructions to read. You can select what type of audio you prefer. Deselecting any of the options will mute that option. Click Continue to start your mission. Using the lab pad is an important way to get the most out of this simulation. Is this the first time you are playing a Labster simulation? Perfect, then let's get started with your first mission. Thanks for joining our research group. Did you know that yew trees are poisonous? I have heard that even horses can die if they eat the poisonous twigs, but also that a yew tree compound called Taxol is used in cancer therapy. This makes me wonder how Taxol affects human cells. I need your help to find out today. Cell division is essential for any living organism. For which of these processes is it not absolutely necessary? Precisely, cell division is required to build and maintain complex organisms, including humans. It is the key process in reproduction, tissue renewal, growth and development. Single-celled organisms reproduce via straightforward cell division. In many higher organisms, this is a little more complex. Meet me at the fluorescence microscopy workbench to get started. Cells are the building blocks of all living things. But did you know that they can be grown in test tubes and petri dishes in the lab? This is a convenient way to study the processes in the cell or test the effect of drugs. I have treated some human cells in a test tube with Taxol to study the effects. The cells have been exposed to Taxol for 24 hours. I also prepared a control sample that was not exposed to Taxol, but otherwise treated the same. Finally, we have samples from before the treatment. The cells have been labeled with fluorescent markers, so we need to use the fluorescent microscope. You will see your cells on the screen. Compare the experimental and control slides found on the workbench by placing each on the microscope. Based on your examination, what is your conclusion? Excellent observation. You can see that after 24 hours the control cells have divided and the number of cells has increased. Conversely, cells exposed to Taxol did not divide much after 24 hours. This demonstrates that Taxol inhibits the cell division. Great job! Cell division is a complex process, so let's find out a bit more about it before we plan further experiments. Click the Diagrams button to see an overview of the cell division process on the screen. Click on the various parts of the cell cycle on the screen to learn about each phase of cell division. G1 phase is the first gap phase. This is a resting phase during which the cell grows and waits for signals to divide. Signals to divide activate the first checkpoint. In S phase, the DNA is replicated. In the second gap phase, G2, the cell grows and prepares for mitosis. The cell is checked for errors and only proceeds past the checkpoint if the DNA has been replicated accurately. G1 phase, S phase, and G2 phase together are called interphase. M phase is divided into a series of phases: prophase, prometaphase, metaphase, anaphase, and telophase. Another checkpoint here helps ensure the chromatids get separated correctly. Perfect. You've learned about the phases that make up the cell cycle. Let's have a closer look at the meiotic phase. There are 3 checkpoints in the cell cycle. What is their role? Yes, these checkpoints are important for ensuring proper division and eliminating the potential of mistakes. Great, let's head to the sample preparation and light microscopy workbench now. When cells divide, the DNA needs to duplicate. To understand how this can happen, it is important to know how the DNA is organized in the cell. Would you like to dive into the cell and find out for yourself? There is a blood sample in the kidney dish on the workbench. If you prepare the microscopy slides this time, I will show you what the inside of a cell looks like. If you need help at any point, click on the lab pad for more instructions. Use the syringe full of blood to add a drop of blood to the microscope slide. Put the syringe down and place a cover glass on top of the blood sample. Place the slide on the microscope stage so we can dive into the sample. Blood is composed of different cell types. Red blood cells are highly specialized transport cells that do not contain a nucleus and cannot duplicate. Red blood cells develop in the bone marrow and circulate in the body for 3 to 4 months. White blood cells, conversely, are much longer lived and are able to duplicate. White blood cells contain organelles that keep the cells alive, just like our organs keep the whole body alive. When a cell divides, both daughter cells need to contain all the organelles. The nucleus stores all the information for building the cell's components. This information also has to be duplicated before the cell divides. The information for building the cell's components is stored in the chromatin, a complex mix of proteins and DNA. Chromatin is usually spread out in the nucleus, but right now, it is packaged into chromosomes. When a cell is about to divide, chromatin condenses into chromosomes. Each chromosome consists of 2 duplicate chromatids. Packing the DNA into chromatids is essential for cell division. Think of chromatids as 2 rolls of tightly wound thread, versus the thread being randomly spread out all over your room. The DNA thread is further packaged around protein complexes called histones, which are stacked into structures called nucleosomes. Now, let's have a look at the DNA itself. As you already know, DNA consists of 2 strands of molecules that form a twisted double helix. Each strand is composed of several million building blocks called nucleotides. This 6 billion nucleotide long code contains all the information for copying and maintaining the cell. DNA wound around histones forms a structure that looks like beaded string. What is this structure called? You are right. Chromatin is a complex of DNA and histone proteins that form the chromosomes. To visualize chromatin, think of a long DNA strand wound around small nucleosomes, like a string of beads. As you know, DNA packaging in human cells is very complex. During cell division, all DNA inside the cell must first be copied accurately. When the two copies are ready, they have to be pulled apart into two genetically identical daughter cells. Finally, the cells physically separate. Together, all of these steps are called the cell cycle. The cell contains two sets of chromosomes. What is the correct name for such cells? Precisely, any cell with two sets of chromosomes is called a diploid cell. A cell with only one set of chromosomes is called a haploid cell. In humans, all the cells in the body, called somatic cells, are diploid. Only reproductive cells, called gametes, are haploid. Once the DNA is replicated, the cell can start the actual division process. To study cell division we can use plant roots and shoots. These tissues contain a region called the meristem that undergoes rapid and continuous growth. The high rate of cell division of this region allows the examination of various stages of cell division at the same time. Use the tweezers to isolate an onion root tip. Place a piece of the onion root into the tube containing hydrochloric acid. Now we have to wait for 12 minutes to make sure the hydrochloric acid penetrates the tissue fully. Luckily, we are in a virtual lab so we can simply speed up the time and continue right away. Transfer the root tip from the tube with hydrochloric acid to the tube with carnoise fixative. Put the tweezers back. Then, place a microscope slide in the microscopy slide tray. Place the root tip on the slide. Put the tweezers down to cut the root tip on the slide using the scalpel. Now, put the scalpel back and use the pipette to stain the root tip with carbylfuchsine. Now, add a drop of water on the root tip sample. Well done. Now cover the slide with a cover slip and put it under the microscope. Examine the onion root tip at different magnifications by clicking on the different objective lenses. First, the chromosomes condense. This is called prophase because it's taking place before the real action, the division. The next clearly distinguishable phase is called metaphase. During metaphase, the chromosomes line up along the equator of the cell because they are pushed there by the cytoskeleton. After metaphase, all the chromatids are attached to the spindle apparatus and then the sister chromatids are separated and pulled apart. This is called anaphase. The onion root tip cells can only divide through the process you just studied. This is called mitosis. The two daughter cells are genetically identical and have the same number of chromosomes. Most human cells also undergo mitosis, but some cells divide via meiosis instead. What is stained in red? You got that one all right. The chromosomes are stained in red. All right, go to the interactive poster near the entrance to the lab. Complete the poster by clicking on the empty fields on the poster and then click on the blue buttons at the bottom of the poster to place this text in the field on the poster. Correct. Fill in the next blank space. The homologous chromosomes, that is the paternal and maternal version of the same chromosome, pair up during prophase of meiosis I. They exchange parts of genetic information, thus creating new, unique chromosomes. Later in meiosis I, each daughter cell will inherit one of each pair of chromosomes, but it is random whether this is the maternal or paternal copy. Correct. Fill in the next blank space. Because in meiosis the cells undergo 2 rounds of cell division, but only 1 round of DNA replication, each daughter cell has only 1 copy of each chromosome. This is called a haploid cell. When haploid reproductive cells fuse during fertilization, the new cell has a pair of each chromosome again. What would happen if humans produced reproductive cells through mitosis instead of meiosis? Excellent. If gametes were produced via mitosis, they would have the same number of chromosomes as other cells, 46. During fertilization, two gametes fuse, so the number of chromosomes would double in each generation. Unicellular organisms such as bacteria depends on asexual reproduction. Why is sexual reproduction so common in higher multicellular organisms such as humans? Very good. Thanks to inheriting half of the DNA from each parent, meiosis ensures that no two organisms have exactly the same genome. The only exceptions are identical twins. Genetic diversity in the population means that the population as a whole is less vulnerable to genetic diseases and more able to adapt to changing circumstances. Remember the slides you examined using the fluorescent microscope? These cells were undergoing mitosis. How would paclitaxel affect cells that undergo meiosis instead? Good thinking. In both mitosis and meiosis, the homologous chromosomes or sister chromatids are pulled apart by the microtubules of the spindle. This means that paclitaxel inhibits both mitosis and meiosis. Great work. You know your mitosis from your meiosis and your diploid cells from your haploid cells. Great job. Now we know that the poisonous compound extracted from the yew tree can stop cell division. Our research group studied this poisonous compound further and found out that it stops the microtubules from pulling apart the sister chromatids. As you remember, this will trigger a checkpoint so that the cell cannot complete the division process. This leads to cell death and can be dangerous. In some cases though, cell death is very useful. Cancer cells divide uncontrolled. Doctors can use paclitaxel and drugs like it to kill these cancer cells and treat patients. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.